Chapter 4, Care and Use of the Microscope. So today we're going to talk about the principles of the microscopy and the different types of microscopes that there are, how to care for the microscope, how to troubleshoot the microscope, and a little bit about the other types of microscopes that might be used in the lab. So the principle of microscopy, the image is on the bottom, okay? The light shines up from the bottom through a condenser up to the specimen. It actually flips that specimen upside down and backwards. So if you were to take something like a little letter E out of a newspaper and put it under the microscope, when you look at it under the microscope, it's going to be upside down and backwards, which seems a little bit strange, but it does flip the image, comes through an objective lens and up through the eyepiece to your retina. So the light is shining up from the bottom up to the top. The eyepiece or oculars are the part where you look through. This can be monocular or binocular. Binoculars are something that you've heard of because that's what you use to look at the birds up in the trees, right? Bi meaning two. If you think back to maybe your high school biology class, you may have used a monocular where there was one eyepiece that you looked through versus the binocular. A lot of students do struggle initially using the binocular. They start to complain, they see their eyelashes, that they can't look through two things at once. I recommend patience. Over time, you will not notice that anymore. Initially, it may drive you a little bit crazy, but bear with me. By the time we get to advanced hematology and microbiology, you guys are going to be complete experts in the microscope, and it will not be confusing to you. There is a power of magnification in the oculars as well. Usually, in most cases, for the medical laboratory microscopes, the magnification of the oculars is 10x. So if you were to take all of the other objectives off and just look through the eyepiece, it would increase things 10 times. We also have interpupillary control. This is the lateral separation of the two eyepieces, which are adjusted by using the interpupillary control. So what this means is if you've got eyes further apart, you can pull them further apart. If your eyes are closer together, you can push them closer together. As you can imagine, this is incredibly important because not everybody's face is the same size. So you will be able to move those in and out, okay? There's an optical tube that you'll see runs from the oculars down towards the objectives. You've got a neck or arm you can hold onto, a stand, and a nose piece that houses the objectives. So what the heck am I talking about? This right here. This is very similar to the ones that we are going to use. So you do have to be able to label this entire thing. Understand what these things are, because there may be times in class where you're saying, I can't find my image. And I'll say, did you move your condenser down? Did you adjust the rheostat? And if you're like, whoa, I don't know what you're talking about, you will if you memorize these names, okay? The eyepieces on top, which hold the oculars. The interpupillary controls where you can move those back and forth. The optical tube is what brings the image from the objectives up through to the eyes. The neck or arm and stand is what you can usually hold onto when you carry it. Okay. The nose piece houses these objectives. There's going to be a few of them we'll talk about in a minute that have different magnifications. The stage clips here hold the slide in, which sits on the stage. The condenser can cause the light to be increased or decreased. Field diaphragm does the same in a different way. The coarse and fine focus is very important. Okay, we're going to have to use those in a certain order. And when we do the laboratory on the microscope in the next week, we will be very, very meticulous with those and how we use them. You can't just go take the coarse one and start moving things around, or you could crack your slide. The light source, like I said, comes from the bottom. We've got a stage, X and Y adjustment knobs, which means it takes your slide and moves it front and back and side to side. The rheostat turns your light up and down. We usually have a power switch. Now, this is not the exact microscope we're going to have, but it's very similar. The power switch will be very close to the rheostat in almost all situations. So the objective power, these are the ones that hang down, and I'll go back for a second, over the slide, okay? Now we have low power and high power. In most cases, that's what we're using in hematology. Low power is usually a 10x. So if you have your oculars at 10x and your objective is 100x, it would be a total of 1000x. If your ocular is 10x and you're using the 10x objective, it would be 100x. So you multiply the ocular times the objective to get the total magnification. In the hematology laboratory, when we are doing blood counts, okay, differentials, we are going to use oil immersion, which is usually 50 or 100x. 100x is usually what we're going to use. The condenser has to be all the way up so we have maximum light, and we are going to be looking at those cells at 1000x magnification. There are a couple different types of lenses, achromatic or panchromatic. Chromatic, usually there's a spheric surface on the lens which acts like a prism, and this is okay unless you're using oil. When you're using oil, the oil can get all up in this spheric surface and dry on there. I think of it as like if you have an old cookie sheet that you've been adding oil and stuff or French fries to for a long period of time, you get that that hard vegetable oil that just hardens over time. That's what's going to happen up in that spheric surface of the lens. These are great for dry type of uses, but when we get to the panchromatic, that's pretty much what we're going to want to use because it's nice and flat and it's easy to wipe the oil off. Oh, here we go. So you can see here a couple different options. The panchromatic, it's expensive, but it's got a flat field. And the achromatic objective lens does not correct for the curvature in a round field, and the edges of the field appear fuzzy and out of focus. So we want to use a plan achromatic objective lens in most cases. There are some other things we want to look at with these lenses. We want to make sure we've got parfocal ability. What that means is when you focus on something at low power and then you switch it to high power, you want it to remain in focus, okay? If you go from low power and then switch to the high power objective immediately, you don't want to have to refocus everything that you just spent your, couple minutes focusing on. You want to be able to go from magnification to magnification without having to refocus. That's parfocal ability. The next term is resolution. Resolution is the ability to distinguish between two objects that are close together as two distinct entities. So it makes an image sharper or clearer, but does not affect the resolution. Parcentric means that an object in the center of field using one lens will be close to the center when you move it to another objective. So if you're looking at a cell, okay, and you're like, ooh, I wonder if that's a blast. Okay, you guys will know what that means in advanced hematology. So you switch your focus from the 10 objective to the 100. You hope that object is still in the same spot. You don't want to have to fish around for it again. Parcentric means it will remain in the same spot. Okay. The focus controls sharpen the image. We've got the coarse adjustment and the fine adjustment. Coarse moves it really fast. Fine is just for that last minute fine focus. The condenser is the aperture diagram, so that can focus the light in a smaller or larger area. And the field diaphragm controls the amount of light coming through the slide. A lot of times when we do a blood smear, we need to have that all the way up and max amount of light. When we're doing something that's a wet preparation that isn't a stained dark slide, we're going to want to turn that diaphragm down. So for example, in your analysis, we're going to turn it down. Hematology, we're going to turn it up. You're going to see me talking about that over and over and over again. So some components and parts, what the heck do all these numbers mean on the objective? I cannot tell you exactly what every single one of these means, but the most important part on here is the magnification, the 10x, or the 10x, 40x, 50x, 100x. That is what you need to be aware of. All of these other ones just kind of come into play if you are a pathologist or you're starting to get into a lot more microscopy, you might care a little bit more. So here's the cover slip thickness. It recommends a 0.17. The working distance away from the slide would be, 0.5 millimeters, it might say here. It's got a numerical aperture. I could not tell you exactly off the top of my head what that number means. Feel free to look that up. It's not something that I look at a lot when I'm looking for an objective. The most important thing that you need to know is that this 40x, okay? Because when you're doing a urinalysis, you're going to want this 40x, okay? Hematology, you're going to want to say 100x. So that's the number that you really care about. But there are going to be other numbers on there, so I just want to point it out that there is a purpose for them. We use bright field illumination. Okay, this is where the light source is focused at the specimen. It's a Kohler system where the light source and condenser are properly aligned. It results in very evenly distributed brightness. So when you look under the scope, you're going to see the whole entire field being bright. It's usually a tungsten type of light bulb. And we have a control knob or a rheostat that can turn the light brighter or make it less bright. So the operating procedure with Kohler illumination, the first thing that we do, and we're going to do this step by step in class, okay? So don't feel that you have to memorize this, because once you do it a few times, it's going to be embedded in your brain, I promise. You turn on the light, you adjust your oculars, you focus on the object, okay, using the low objective. You position the condenser to the highest position because we're in hematology. Adjust the diaphragm for light. Then you're going to move to the oil immersion, okay? After that, you remove the slide and you're done. So those are the steps, and we're going to do them in great detail together as a group. All right, we have different types of immersion oil. The purpose of the immersion oil is very important. Increases the refractive index when the 50x or 100x is used. You do not ever use oil on the 10 or 40x. We have different types. So type A is very low viscosity. We can use that in fluorescent and dark field microscopy. B is high viscosity. That's what we're going to use. And very high viscosity is used for inclined microscopes or scopes with a long focus objective lens and wider condenser gaps. So viscosity means how thick is it, okay? So the procedure that you would use for that is once you get the oil objective under the scope or close to it, you're going to add a drop and then you're going to dive that objective into it. Don't panic right now if you don't know what the heck I'm talking about, because we're going to do it in class together. When you're not using the microscope, we're going to keep these protected from dust. The new microscopes that we have in the classroom are very, very expensive, and I'm very excited to use them. They're the nicest ones that I have used in a classroom by far. Before we use it, we want to inspect it. Make sure it's clean. We want to transport it as this lady shown here. You want to hold a hand under the base and one on the neck and the back. Be very, very careful when transporting. You do not want to drop something this expensive. Basic troubleshooting is on page 58 of your book, but sometimes you're going to find issues with not being able to focus. A lot of this comes in time with practice. And a lot of this comes with us working together and doing these things together. Some of the more common things that I see are students putting the slide in upside down. Okay, that's a big one. Or they are not focused in the right plane. Okay, they're looking at the opposite side of the slide. So if you focus into the wrong part of the slide, you're not going to see the right thing. I see this a lot in hematology. Other types of microscopes used in the clinical lab. A few of these may be used, some of them are not used very often, but phase contrast microscopes look at the thickness of the object. So if you want to do a more exact platelet count, you see there's little halos around them. They may be easier to see. We usually don't have too much of a problem looking at those because we have a good trained eye. Polarized light microscopes, we use this for things like urine crystals. Okay, we don't have polarized light microscopes in our class, but we're going to look at what does it mean to have these blue and yellow crystals in here. Okay, they could mean monosodium urate. They could mean a couple other different types of crystals could be present. Those are all really important when it comes to not just urine crystals, but also joint crystals. Does somebody have gout or do they have something else? So this is a way that we would determine that. Dark field microscopes, light is picked up by the object. So for example, spirochetes and syphilis, they actually look like this, okay? It's caused by Treponema pallidum. It's the syphilis agent, and it's like a corkscrew, okay? They're almost impossible to see on gram stain. They're so tiny, but if they're like this, it would make it a little easier. Even with this, very, very difficult. We usually have to use other types of testing methods to see these. And then a fluorescent microscope uses fluorochrome dyes to be able to see the actual bacteria or virus-infected cell glow. All right, that concludes our section on microscopes. I look forward to using these in class with you guys within the next week.